Our next presenter is Dr. Elli Papaemmanuil, who is at Memorial Sloan Kettering Cancer Center. She's a molecular geneticist who looks at systematic interrogations of well-annotated clinical trial cohorts in leukemia and other cancers to explore the compound genetic and clonal architecture in patients' leukemias and their tumors to help dictate the clinical phenotype, disease progression, and clinical outcomes. She works with a great multidisciplinary team there at Memorial, and she is going to talk to us about the impact of oncology therapy on clonal hematopoiesis, which we heard a little bit about earlier, and then the subsequent risk of developing a therapy related to leukemia in cancer survivors. So Dr. Papaemmanuil, welcome.
Hello, thank you so much for the invitation and the opportunity to be here today and present on behalf of our team at Memorial Sloan Kettering Cancer Center on a very important topic that has been enabled by the V Foundation. And I would like to say that it's really inspiring to hear the talks today and to see that so much research that is being supported that extends beyond the realms of how we can optimize, not only how we can optimize primary cancer care, but most importantly, how can we think holistically about our patients and how can we account and improve the treatment, taking into account comorbidities as well as long-term risks. So very, very appreciative of your support. So a major foundation on our understanding of cancer development, at least from a molecular perspective, is that cancer is very much the end result of a stepwise acquisition of acquired mutations. So what happens is a normal cell will often acquire a mutation that will give that cell fitness over other cells, and it would allow it to expand as a uniform cell population. And what we do understand is that in the vast majority of cancer types, there is a stepwise acquisition of mutations that parallels very much the principles of Darwinian selection that results in populations of cells that have acquired mutations that can invade their local environment, resist treatment, and ultimately result in metastasis. So in recent years, molecular profiling studies have allowed us to understand that for all of us, as we live and breathe, our probability of acquiring these mutations across any tumor or any tissue type in our body increases over time. And usually those mutations really do not have any consequence. But if they do hit the right gene in the right cell at the right time, then they will lead into this first clonal expansion. So in recent years, there's been studies that focus on sequencing normal tissues in order to understand how these processes happen, and most importantly, understand what other potential exposures or environmental parameters may stop a cell with a particular mutation from growing and allow another cell with a particular mutation type to expand. So these are parameters that may increase selection and fitness of particular cells. Now, as we heard earlier, the acquisition of mutations and the establishment of a clonally expanded population in the blood is referred commonly to as clonal hematopoiesis. Studies in the blood have been the most advanced because it's very easy to sample blood. So we're starting to understand this phenomenon very well. And population studies by Ben Ebert groups, as well as others, have shown that the incidence of clonal hematopoiesis increases with age. And if we look at this incidence curve over time, it very much reminds us of the incidence of cancer. And in fact, both cancer and clonal hematopoiesis have this shared age-related incidence, with the peak really being at individuals of the age of 60 or 70 years old and greater. So we asked the question as to what is the impact of CH in cancer patients. And this is important because we know that a major complication or cause of mortality following treatment in cancer survivors is the development of therapy-related myeloid neoplasms, or TMN, as indicated here. And therapy-related myeloid neoplasms constitute 10 to 20% of all primary myeloid leukemia diagnoses. They affect about 0.62 patients per 100,000 patients, and the survival is actually very dismal. And in fact, therapy-related leukemia is considered to be one of the most lethal cancers amongst all cancers. Here we see that the incidence of therapy-related leukemia can vary by treatment type, but the majority of the survivors, for example, the largest proportion of the absolute number of patients with therapy-related disease come for some of the most frequent cancers, such as breast cancer or non-Hodgkin lymphoma. And this is important because as we get better at treating the primary cancers of patients, we now have about 13.7 million cancer survivals in the United States, and this is expected to rise to 18 million. So I should also say, historically, it was thought that oncologic therapies such as chemotherapy and radiotherapy during primary treatment caused additional mutations or new mutations in the blood, and that those mutations led to the subsequent development of therapy-related leukemia. However, as we appreciate the phenomenon of clonal hematopoiesis and the fact that individuals with clonal hematopoiesis in normal populations are at an increased risk of developing myeloid leukemias, we try to ask the question as to what is the effect of oncologic therapy in clonal hematopoiesis in determining the risk of therapy-related leukemia in cancer patients? And in many ways, we were uniquely positioned to ask this question because at MSKCC, every month, about 800 to 1,000 new patients who get onboarded into our hospital will be consented for tumor profiling using our in-house assay, MSK IMPACT. And part of that diagnostic process is to sequence the tumor as well as the blood in order to provide the oncologist with a report for the mutations that are present in the tumor of that patient in order to inform optimal cancer care. However, part of this diagnostic workflow means that data across a panel of just under 500 genes is also being generated for the blood, and that very much gives us the opportunity to go and study clonal hematopoiesis in our cancer patient population. So the study, generously supported from the V Foundation, allowed us to look at the first 24,000 or so patients that have been sequenced with MSK IMPACT, of which 10,000 patients we were in a position to extract very detailed treatment, demographic, as well as behavioral information such as smoking. We showed that 1 in 4 patients in our center are actually carriers of clonal hematopoiesis at the time that they come to receive treatment. And then we asked the question whether clonal hematopoiesis is more frequent in patients that have received prior treatment, say those that may have been referred from another hospital. And indeed, we found that clonal hematopoiesis was more frequent in patients that had received prior treatment. But what was more striking, shown here in orange bars, was that patients that had undergone oncologic therapy seem to have clonal hematopoiesis with a very particular type of mutations, such as mutations in P53, CHEK2, or PPM1D. And this is important because these genes are part of a machinery in our cells that make sure that we repair DNA damage. What our ability to link very detailed treatment data with our molecular data allowed us to see that, or to find that clonal hematopoiesis was more frequent when patients had received particular types or classes of chemotherapy. For example, even amongst platinum agents, we found that patients who had received carboplatin as opposed to oxaliplatin had a lot more clonal hematopoiesis than the ones that didn't receive this type of therapies. And in fact, we also identified that there were dose-dependent effects, whereas the more treatment a patient received, the more likely the risk of being CH carriers for those patients. We also were in a unique position to obtain serial samples during the clinic primary care for those patients for approximately 500 patients. And these were distributed between patients that didn't receive therapy between the two sampling time points and patients that had received an array of these different therapeutic regimens. And here we show the dynamics, the changes of the number of cells that may carry those mutations in the blood between the two time points. And we indicate in red when those cells expand, in blue when they contract, and in gray when they remain stable. And what we identified was that in patients that received cytotoxic therapy and radiation therapy, there was a lot more expansion of those cell populations in the blood whilst they were undergoing the primary cancer care therapy. And in fact, when we studied the type of the mutation that may have defined those type of cell populations, some patients may have had mutations in, say, in genes such as DNMT3A, TET2, which are very frequent in the population, and in fact the most frequent genes in clonal hematopoiesis. When we compared whether those cell populations expanded or not during therapy, we see here that there is no difference between the blue boxes and the orange boxes. However, in the patients that had clones or cells with mutations in one of those three genes involved in DNA damage response, the individuals that received therapy in orange in between the two sampling points expanded at a much greater rate than the ones who didn't receive therapy. So this shows that patients that may have had mutations in these genes not receiving oncologic therapy kept those cell population more in check or at more stable levels than the ones who did receive the therapy. So this allowed us to add a layer of our understanding in that therapy-related leukemia may not be necessarily directly caused by mutations that as a consequence of treatment, but in fact that it could be a result of a selection and an expansion of cells that already exist at the time of diagnosis. And then following subsequent additional genetic and clonal evolution, those cells go on and develop into the therapy-related neoplasm. This, of course, allows us to understand what are the critical pathways that define those cells. And in fact, the fact that we understand that now the cells that go and seed this therapy-related leukemia are defined by components of the DNA damage response machinery allow us to understand that the reason this tumor, this leukemia is so lethal is because the very genetic makeup of those cells expands and thrives in the presence of treatment. And it allows us to explore therapeutic avenues that target that particular biological pathway. However, beyond targeted therapies, we then ask the question whether this knowledge, given that we may be able to identify which patient at the point of care may be a clonal hematopoiesis carrier, is there anything that we can do in order to decrease the risk of this subsequent leukemia? So we modeled what are the risk factors that might predict for subsequent development of therapy-related leukemia. And we found that the presence of CH, the number of mutations, and most importantly, the type of the mutations, such as mutations in P53, are strong predictors of subsequent development of therapy-related leukemia. So with this in mind, we tested a scenario where now a patient at presentation, given example of this patient is a 72-year-old female, presents at MSK with an early stage breast cancer and is discussing therapeutic options with her oncologist. One of them may be a more a therapeutic protocol that has less treatment intensification and a wait and watch approach where surveillance may be implemented in order to check for recurrence of breast cancer. The alternative option is to increase the intensity of oncologic care to ensure that there's no recurrence of this breast cancer. However, now we know that this patient has a p53 mutation in the blood. And we know that if we were to administer intensive therapy in this particular patient, we might accelerate that clone in the blood and might increase the risk of developing a much more lethal disease than potentially the recurrence of an early stage breast cancer. So we modeled this in a breast cancer patient to see if we were to develop risk-adapted treatment decisions at the time of primary care, is there an opportunity to decrease the likelihood of developing therapy-related neoplasm down the line for the patients most at risk? And in fact, what our model showed was that in the patients most at risk of therapy-related neoplasm, we could decrease that risk by 8% without affecting their overall likelihood of survival for their primary cancer. So this sets a framework for the design of future prospective clinical trials to allow us to formally validate this inference and test it. But at the same time, at MSKCC, we formed one of the first clonal hematopoiesis clinics where we can identify patients at high risk with CH, at high risk of therapy-related disease, and bring them into the clinic and offer surveillance protocols and work with our teams in order to understand this better. So with that, I would like to thank the patients and the families, the V Foundation, for being the primary supporter of this research project that has now led to many more projects within MSKCC. This is really a true representation of many centers from our hospital, and Kelly Bolton, who drove this study, as well as everyone else in the group and in the lab. Thank you so much.
Thank you so much, Dr. Papaemmanuil. That's, again, excellent work, and we're delighted to know that it is leading on to further studies. A huge accomplishment. Thank you.